Hello and welcome to our webinar. I'm Adam Elwan, your host today. So to get us started, these are some of the things you can expect. You'll be gaining a better understanding of the information requirements and risk assessment for the approval and authorization of in situ generated active substances as well as their products. Our presenters will also be going through the information requirements, methodology, and decision schemes that need to be followed when doing your risk assessment. Last but not least, you'll also have the opportunity to ask questions from our live panel of experts who've joined us today, so do make use of that. Now, before we get started, a quick note on how to ask your questions. Join the Q&A on Slido, either by scanning the QR code on this slide or going to slido.com and entering the event code INSITU25. You can send your questions throughout the event, and we'll keep the Q&A open until 1 Helsinki time, that's 12 CET. To help us stay focused, please keep your questions within the scope of today's topic. Your questions will be replied to during the live Q&A, where our panel of experts will be replying. If your question is not answered live, we'll publish a Q&A with responses afterward. And if something comes up after the webinar, you can always reach us via our contact form at eca.europa.eu/contact. All of today's materials will be made available on our website. You'll find the video recording as soon as the broadcast ends, and you can already download the presentations. So if you miss anything or want to revisit a section, everything will be there for you. Here's the program. We'll start with Eva from ECA, who will introduce you to the recommendations and will give you general information. Then Andreas Uphoff from ECA's AP-CP team will go deeper into the analytical methods and physico-chemical properties that need to be covered. After him, Mari from ECA will then take you through the efficacy requirements. After her, we have Isabel Kriegel from the Austrian Authority, who will talk about the common elements for human health and environment, and will follow with the toxicological information requirements. The last presenter today is again Eva, who will come back this time to talk about the information requirements for environment and their assessment, and she will be followed by the conclusions from the webinar. Let's get started with our first presentation. Eva will tell us what are in situ generated active substances and their products. Eva, the floor is yours.
I would like to start with a generic introduction about the in situ generated active substances and their products. My name is Eva Valkovičová and I work in ECA in the biocides unit dealing with active substances. I will explain what in situ generated active substances are and how they differ from the conventional substances. We will see which types of products linked to in situ generated active substances exist, and I will conclude with the nuances for active substance and biocidal product assessment of the in situs. Let's begin with a short introduction to the world of biocides. Before a biocidal product can be placed on the market, the active substance needs to be approved. The approval is granted per product type, and it needs to show the hazard properties and efficacy of the active substance. It also needs to prove that at least one biocidal product is safe to use. After that, the technical equivalence comes to play. In this process, it needs to be proven that hazard of an active substance produced in a different way or elsewhere is equivalent to the approved substance. And finally, in the biocidal product authorization, only products with approved or technically equivalent active substance can be authorized. And in this process, it needs to be shown that each biocidal product can be used safely. Therefore, in the conventional world, first, the active substance is produced and then it is assessed. After that, the substance is used to formulate biocidal products. So the basic assumption is that there is no change in the active substance after its manufacturing and before its formulation into a product. But what if the biocidal product exists before the active substance? What if the active substance is generated in situ? Here you can see the in situ generation system. The active substance can be generated from one or more precursors by mixing, by using of a device or a substance or a mixture that contains a catalyst. As you can see, the chemical composition of the in situ generated active substance will therefore depend mainly on the precursors used or on their concentrations. Let's take as an example in situ generated peracetic acid. We have 3 different in situ generated active substances. The pure active substance is always the same, peracetic acid, but the precursors are different, and therefore also the name of the substance is different. So the substances you can see in the blue boxes are the different precursors, and you can note that in the first 2 the precursor is tied and it is the same, while in the first and the third blue box the precursor hydrogen peroxide is the same. But still, these are 3 different active substances generated in situ. So what are the differences between the conventional biocides and the in situ systems? It is, in fact, that the products usually exist before the active substance, that the active substance is usually generated from a product, the manufacturer and the place and the method of manufacture pretty much varies, the naming convention is different from the conventional one, and that is because in the in situ generated active substance, the name bears the name of the precursor, and sometimes also the method of manufacture, like, for example, by electrolysis, by acidification, by oxidation. Then the analytical profile of the in situ generated active substances we call global composition. The technical equivalence is not done for the in situ generated active substance as such. However, the technical equivalence is relevant for certain precursors, and the manufacturing process, of course, needs to be always controlled. Let's dive deeper into the technical equivalence. In the technical equivalence dossier of the in situ generated active substance, you need to submit the information about the precursors, which are marketed as biocidal products, unless it was agreed otherwise in the approval process. This, however, also means that as a prerequisite, the reference specifications must be set for these precursors marketed for biocidal use. And this means that the 5-batch analysis would have needed to be provided, most probably provided in the active substance approval stage for such precursors. The technical equivalence is not required if the precursors are not marketed for biocidal use. However, these precursors should meet international standards. The technical equivalence is also not needed for active substances generated in situ from non-marketable precursors. So, for example, for air or water. To conclude the part of the in situ generated active substances, I wish to highlight that the same rules as for any other active substance shall be followed, and the assessment shall comprise the active substance, the solvents that cannot be removed, the reaction byproducts, and other impurities. What is important is, of course, to have the representative biocidal product. But what is different is that for in situ generated active substances, also information and assessment of the precursors is needed. And now we are moving towards the biocidal products linked to in situ generated active substances. I say linked, as obviously there are no in situ generated biocidal products. However, we sometimes do call them the in situ products. What is important to know is that there are 4 types of in situ generation systems. And even more important is to realize that for the case type 1, 2, and 3, the biocidal product is the precursor. And then for case type 4, the biocidal product is the in situ generated active substance. I will now introduce you to all 4 cases, to all 4 case types, and I will provide you also with examples. The case type 1 is when the in situ biocidal product involves a generation system based on mixing of 2 precursors or more, and one of the precursors can be a commodity chemical. In this type, this precursor marketed for biocidal purposes, or the precursor not marketed for biocidal purpose, which we sometimes call also the commodity chemical, are the biocidal products. As an example, I choose peracetic acid, but this time generated from the same as we have seen before, so from TAET, but from the second precursor being sodium percarbonate. Peracetic acid is marketed as a broad-spectrum disinfectant that kills bacteria, viruses, and spores. This example of the in situ generation system is a disinfectant detergent for laundry, and it is in the form of tablets containing the precursors. So the 2 precursors, TAET and sodium percarbonate, are both as a solid compounds, and they are together in a tablet. When this tablet comes into contact with aqueous media, then the TAET reacts with sodium percarbonate, which is the source of hydrogen peroxide, and peracetic acid is generated. The next case type is case type 2, and the in situ biocidal product involves a generation system based on 1 or more precursors marketed as a biocidal product used in a device. One example is ozone generated from oxygen, but when generated from oxygen bottles marketed for biocidal purposes. Another example is active chlorine generated from sodium chloride by electrolysis. Here, the example of the in situ generation system is sodium chloride used in a system which generates the active chlorine to treat wastewater of public flushing toilets. We have 1 precursor, sodium chloride, and 1 device, an electrolysis reactor. After a bacterial treatment and a sedimentation step, the remaining liquid passes through the electrolysis reactor, where it is converted into sterilized water and finally is reused for flushing the toilet again. Case type 3 is when the in situ biocidal products involve an in situ generation system based on any substance or a mixture containing a catalyst that, when exposed to a mode of activation, generates free radicals out of air and or water present in the immediate environment. For this case type, the example we have is free radicals generated in situ from ambient air or water. Here, the in situ generation system is a self-adhesive film which is consisting of a polymer containing zinc oxide as the catalyst. By catalysis with air, humidity, the zinc oxide will generate the reactive oxygen species, and this species will disinfect the area. This film can be applied on different types of surfaces, on tables, on doors, or on handles. Now moving to the case type 4, the in situ biocidal products involve a generation system generating the in situ active substance from precursors or from formulations containing the precursors that are not placed on the market for biocidal purposes. And there is a device being used, or the precursors are mixed together. As an example, for the in situ generation system, we can have, for example, ozone generator, which is made available on the market to disinfect and eliminate viruses and bacteria in closed areas. The device is a generator using a corona discharge and generating the active substance, ozone generated from oxygen, where the precursor is oxygen, but this time from ambient air. It is claimed to eliminate unpleasant odors like smoke and any odors from, for example, animals. The case type 4 also covers when free radicals are generated from ambient air and water using catalysts which is incorporated in an object such as tiles or toilet bowls, for example. The in situ biocidal products involve a generation system generating an in situ active substance from catalysts not falling under the case type 3. So, for example, the free radicals generated from ambient air or water using a catalyst incorporated in, for example, a ceramic or a plastic polymer object. Now, after we have seen the 4 case types, let's get to the biocidal product authorization application for the in situ products. Before submitting an application, you need to check if your precursors require a technical equivalence, and if yes, submit it as per active substance. Then, you need to do a similarity check of the output of your in situ system and compare it with the global composition of the active substance generated in situ as it was approved in the active substance approval. And lastly, you need to check the composition of the output of your in situ generation system and see whether there are more data and assessment needed. Now here, you see the graphical example from the recommendations explaining the comparison of the in situ generation system output with the global composition. The global composition, as set in the in situ generated active substance approval, is indicated in red, and the constituents that are part of the and these are the constituents that are part of the in situ generated active substance. In blue, the content of the in situ generated active substance output is shown. So here you can see that the impurity 2 and 3 and the solvent that cannot be removed are within the global composition. The pure active substance and the impurity 1 are constituents of the in situ generated active substance, but are in the output in higher concentrations than they were in the global composition set at the approval stage. The new impurity 4 and 5, as well as the coformulants 1, 2, and 3, and the additional solvent, are the components of the in situ generation system output. Important to remember is that only the constituents already included in the in situ generated active substances at the approval stage, meaning in the global composition, and the pure active substance are considered as part of the in situ generated active substance at the product stage, whereby any deviation from the global composition remains possible, but it could trigger additional data requirements. If the composition of the in situ generation system output is different than the global composition at active substance approval, then this means, as said before, that further assessment of these components that are different may need to be done. And of course, this assessment may require new data to be submitted. To wrap up, the information requirements for the authorization of biocidal products, also for the biocidal products linked to in situ substances, are the same as for conventional biocidal products. And the active substances, of course, need to be assessed. In addition, however, also information and assessment of the in situ generation system output, and then any deviations from the global composition need to be checked. To conclude, what we need to have in mind is that the in situ systems are different from the conventional active substances and products. However, they are part of the biocidal product regulation, and this regulation applies to them. The assessment must cover precursors, active substances, and reaction outcome, and its impact on human health and the environment. Precursors may be regulated under each. For the classification, it must follow the CLP rules. The harmonized classification of as pure as possible active substance is needed, and self-classification of the in situ active substance and of the product needs to be included in the dossiers. I would like to finalize this generic part of our webinar by inviting you to visit our website and certainly to read the recommendations. The document was developed in collaboration with the experts from the Biocidal Product Committee's working groups. Thank you for your attention, and now I will let Andreas to take you through the analytical methods and physico-chemical properties requirements.
Hello, my name is Andreas Uphoff. I'm a chemist, and I'm going to tell you a bit about the requirements for phys-chem properties, physical hazards, and analytical methods for in situ substances. As Eva already told you, the requirements are as for conventional substances, and the waiving possibilities are the same, so we could stop here. But the trick is in what substance we are talking about. We have different things we call substance. Let's start with the most pure, which is the pure active substance, that is the presumed or real driver for the efficacy and the hazard of an in situ generation system. This is the target of classification and labeling, the harmonized classification. If we go one step more complex, we can add the leftover precursor if they exist, impurities from the precursor which didn't react, side products of the generation process. Then we end up with the, if we exclude the solvent, which usually is present, we end up with the dry active substance. Doesn't need to be physically available at any time. And this will be the target of the analysis, because usually in situ active substances are present in very low amounts in solvent, and we would never see anything about the composition of the system if we wouldn't require more. This can be quite challenging, but as a standard requirement, standard waiving possibilities apply. You can usually, if you cannot analyze it, you can waive it for technical impossibility. Next complexity is adding solvent. You know, that the definition of substance is including solvent that is required for the stability of the substance. So the in situ active substance, as the substance we're talking about in the BPR, is including solvent. The agreement is not to insist on the minimal amount of solvent that is possible at all, but just the minimal amount of solvent observed in the generation system in the approval. Any additional solvent present would be considered a co-formulant, and we end up with the technical concentrate. And of course, if you have a product which is including co-formulants like dyes, which do not react in the generation process, this will be also part of the output of the generation system. To repeat, we have the pure active substance, the main active ingredient or constituent, the most potent and driving the hazard of the substance. This is the target for harmonious classification. We have the dry active substance, which excludes any solvent, doesn't need to be physically available. This is the target of analysis. We have the in situ active substance. This is the subject of the BPR for approval, includes as much solvent as necessary for stability of the active substance. And we have the output of the generation system, which includes everything else, including potentially co-formulants. As for conventional active substances, it needs to be ensured that the composition of the active substance in the product finally used is covered by the approval of the active substance. So we need to ensure that the composition is the same, at least in terms of hazard. As Eva has also told you, the active substance doesn't exist independently of the product. What exists independently of the active substance is the precursor. So we cannot do much in terms of control of the active substance before the concrete product is known. So most of this assessment will be done in product assessment. What can be done is to control that the precursor is hazard equivalent and that we know about the precursor. That can be done in technical equivalence, and I'll come to that in a moment again. So most of the control of the active substance is done in the product assessment. Precursors that are not marketable as biocides, which means mostly air and water, then the full variation that can be expected in these precursors and the effect on the active substance composition needs to be assessed in the approval. And if this is too much variation, it cannot be predicted or it doesn't lead to safe active substances, then the variation possible for the precursors needs to be restricted in the use description, which means, for instance, restricting the use of water to drinking water. Also, precursors need to be controlled because you can be exposed to precursors. This is easier because precursors are usually stable substances, so this can be done the standard way via having a reference specification and controlling the hazard equivalence via technical equivalence. On a case-by-case basis, this could also be done by referencing to external standards, ISO norms, EN norms, if they're available and applicable, and compliance with these standards can be shown. For non-marketed precursors, as before, the full variation of these precursors needs to be assessed and concluded that they're acceptable, or you need to restrict the possible variation in these precursors in the approval and in the authorization. To describe what was done in the approval, what was the scope of the approval, we document something we call the global composition, which is just a description of the active substance composition seen in approval. This is for the in situ active substance, as we see before. So it contains the pure active substance, it contains all precursors that's still left. It contains impurities from the precursors that haven't reacted. It contains side products, anything that is important in the reaction. It does not contain coformulants. They don't belong to the active substance. It does not contain disinfection byproducts. The constituents considered relevant will be published in the assessment report for the approval and can be referenced by authorization applicants. The complete global composition may only be known for the evaluating competent authority. If you have the global composition, then you can compare the global composition against the composition generated in the output of your product. And the agreement is that everything documented in the global composition is part of the active substance. And for that part, reference to the active substance data might be appropriate. Anything that is not documented in the global composition, or anything that is documented but is observed at higher concentration, will be needed to be assessed as if it was a coformulant. This is except for the pure active substance if a higher concentration of pure active substance is observed, which can happen because we didn't insist on the absolute minimum amount of solvent. Then this would need to be considered if there is enough data in the approval to support this higher concentration. If not, then approval data might need to be amended. The constituents that need to be assessed as if they were coformulants, we have to know that they're not coformulants necessarily. They might be, but they don't have to be. They'll still be assessed using the same methodology, which includes an assessment if they are substances of concern or not. And another interesting point about these so-called coformulants is that they might not be data-rich. Usually we have coformulants that are added on purpose into the formulation and their properties are known. For these, they might be side products which are not known, and there might need to be some data generation, at least QSAR, or an argumentation why they don't matter if they don't matter. For the active substance, as we said already before, we have the same data requirements as for conventional substances. For the precursor, the same. The waving possibilities need to be considered. The same standard waving possibilities apply as for other conventional active substances and substances, which includes technical impossibility, availability of other data that is appropriate, and so on. We need a self-classification for the active substance, including all side products, impurities, and so on. We need a harmonized classification for the pure active substance, which has the advantage that we have one harmonized classification for all generation processes of, for instance, peracetic acid with different precursors. We need analytical methods for the precursor and the in situ active substance. We do only need monitoring methods for the active substance itself, not for the precursors, except when they're part of the output of the in situ active substance and are considered relevant. For the biocidal product, it is important to have an idea, a proof that the output of the specific biocidal product is in scope of the approval, which means that you need to show that the precursors are in scope of the approval and the whole variation expected in the output can be controlled and is in scope of the approval. The standard data requirements apply also for the product, which includes requirements on the active substance, for which you can rely on active substance data usually, which you should also do if possible. Please reuse the data of the approval in the authorization process. key takeaways: there are many things that we call substances. Different substances are important for different parts of the data requirements. The active substance composition needs to be controlled, but this cannot be done using technical equivalence. It has to be done in the product and via controlling the precursors. The data requirements are the same as for conventional substances, for precursors and active substance and the product. Use the global composition to decide whether active substance data can be reused or not, or if more data is necessary. And waiving is possible in all cases if justified in the same way as for conventional substances. That was it. Thank you.
Hi, I'm Mari Raulio, Scientific Officer at the European Chemicals Agency. I will give an overview on information requirements for efficacy assessment of in situ generated active substances and biocidal products. First of all, as the efficacy assessment of biocidal product is strongly linked to the intended use and the product type, the type of active substance has limited effect on the information requirements. Thus, it's good to remember that all requirements and efficacy criteria set in the BPR guidance apply to in situ generated active substances. In general, efficacy needs to be demonstrated in the use conditions and against all target organism groups claimed for the whole output of the in situ generation system. As a reminder, the whole output, the soup, consists of the in situ active substance, including, for example, unreacted precursors, impurities, and coformulants. The in situ generation system, as well as generation conditions, should be clearly described and then reflected in the test design to mimic the real-life conditions as closely as possible. As for conventional active substances, also for in situ active substances, the efficacy should be demonstrated with data from laboratory tests conducted according to standard test protocols. However, for some uses, such methods do not exist, and then suitable scientifically robust testing strategy has to be developed, possibly by adapting the existing protocols to demonstrate the efficacy of the in situ generation system. The testing strategy may include development of novel methods and design of test setups, which should be agreed with the member state competent authority. In some cases, the efficacy of the in situ generation system can only be demonstrated in field trials under real-life conditions. In such case, all relevant biotic factors should be included in the test setup, and 3 or more replicates should be performed, and an untreated control should be included. Replicates can be tests at 3 separate sites or tests performed at different periods or independent locations of the same experimental site. In situ generation process will not always lead to a single in-use concentration of the in situ active substance. And for some, the in-use concentration cannot even be measured, for example, free radicals. In such cases, as the assessment of efficacy is challenging, it's in the responsibility of the distributor the end user to perform on-site validation and of the system and devices to ensure that the system is efficacious under the use conditions. The biocidal effect of the in situ generation system should mainly be caused by the pure active substance. However, in some cases, the precursors may have some effect on their own and thus contribute to the efficacy, especially if one or more of the precursors are notified as active substances. When the concentration of unreacted precursors is within global composition presented for the active substance approval, they are included in the soup, and thus there is usually no need to provide additional data. However, when the unreacted precursors are presented in an amount which is higher than in the global composition, additional efficacy data on unreacted precursors and their concentrations may be required for the authorization of the biocidal products. The provided data should demonstrate the impact of precursors on the efficacy of the in situ generation system. This slide presents some examples on factors that may affect the efficacy of the in situ generation system in different case types. These factors should be taken into account when developing testing strategy to simulate the intended use conditions. For case type 1, factors possibly affecting efficacy of the system are the in-use conditions such as temperature and pH and co-formants. For case type 2, in addition to in-use conditions, the device and the parameters used in the generation process can have an effect. For case type 3, also other factors might be important, like humidity, stability of the catalyst and in situ active substance, and characteristics of the mode of action, for example, light intensity and wavelength in photocatalysis. For case type 4, all previously presented factors apply. Due to complex nature of some in situ generation systems and respective abiotic factors influencing the generation process and efficacy, no comprehensive guidance can be prepared, and case-by-case decisions may be necessary. In case of a doubt whether the design of the test setup or generated efficacy data are sufficient to cover the intended uses, the member state competent authority and efficacy working group should be consulted. And with that, I come to the end of my presentation. Thank you for your attention.
Hello, my name is Isabel Kriegel. I'm working at the Environment Agency Austria in the biocides team, and I contributed to the development of the in situ guidance. Today, I will reflect on common topics for human health and environment, as well as on specific aspects of the human health assessment. The scope and focus of the assessment of in situ generated active substances, or ESAs, is a bit different compared to conventional assessment. It must address both the active substance generated in situ and also the precursors used in the generation process. But the assessment does not end there. You also have to look at unreacted precursors, reaction byproducts, and impurities, and address them in risk assessment where relevant. So, for the in situ generated active substances, the information requirements of Annex 2 apply. So, this is the same as for conventional active substances. But there are different approaches how to transpose this in case of ESAs. You remember that the composition of ESAs is quite complex and is described in the global composition. There are several single constituents which belong to the ESAs. It's the pure active substance, and then there are impurities like, for example, unreacted precursors, reaction byproducts, solvents that cannot be removed without changing the composition, and other impurities. For example, impurities already stemming from the precursors. So the question is, do we test the ESAs as a whole, or should we test the single constituents? There have been a lot of discussions on this, and we came to the conclusion that both should be possible. So there are now 4 different approaches how you can provide the Annex 2 information requirements. And these approaches are the same for human health and environment. In approach A, you base the data package on the ESAs. As the ESAs has variable compositions, one has to take care that the composition of the tested ESAs reflects the worst case constituent levels. This worst case needs to be well justified, and you also have to consider the stability of the ESAs. So this approach A might not be the best approach for very unstable substances, and it also specifies the concentration of constituents which are tested. Then there is approach B, which is the constituents approach. Here you provide individual information and data on pure active substance, solvents and impurities. The hazard assessment follows SILP mixture rules to assess ESAs using worst-case concentrations from global composition. So those are the basic possibilities. And of course, you might also have a more diverse data set. Therefore, there is also approach C, the mixed approach, where some endpoints are covered with information on the ESAs, while others are covered with information on individual constituents. This approach requires careful endpoint-specific consideration. And last but not least, there is approach D, which is a combined approach. For the same endpoint, you provide information on ESAs and in addition on individual constituents. For precursors, also the Annex II requirements apply. Core data are even necessary for commodity chemicals. However, there might be an increased use of literature data or exploration of waiving possibilities. While these precursor substances are not covered by Article 5, they still require careful risk assessment to ensure safety and compliance. Conclusion on exclusion criteria must be drawn for ESAs, and here the precursors might contribute to classification when they are on hand as impurity which exceeds 0.1%. Last but not least, also waiving principles might apply. When it comes to product authorization stage, you need Annex II and Annex III data in line with Article 20 of the PPR. Usually, the Annex II data set is covered by a letter of access. So what you do first is to compare your in situ generating system output, this is the IGS output, and you compare that to the global composition that has been set at active substance approval stage and check if the IGS output lies within that ranges for all constituents. Next, you identify any deviations. Depending on the data set and the approach which was taken at approval stage, additional Annex II data might be necessary. And finally, you assess the impact of the deviations on classification and risk assessment. Any constituents that are present at higher concentrations compared to the global composition are handled like substances of concern. Constituents already identified as relevant impurities at approval stage automatically qualify as substances of concern based on other grounds of concern. The risk assessment for human health follows the PPR Volume 3 guidance, taking into account appropriate reference values. The assessment is also influenced by the way the ESAs is generated. You already learned today that there are 4 different case types for in situ generated systems, depending on how the system exactly works. Simplified expressed, there might be mixing of precursors, which is case type 1. It might be ESAs generation using a device, which is case type 2, or formation of free radicals with the aid of a catalyst, case type 3. Case type 4 covers special cases with the aspect of non-marketability. This can be precursors not placed on the market for biocidal purposes, either being mixed or being used in a device, or free radicals generated by the aid of catalysts incorporated in an object. With regards to risk assessment, in case type 1, Annex 3 data are required for the precursor formulations. Risk assessment for precursors may use existing evaluations from other frameworks. Co-formulants might be substances of concern that have to be addressed. In case type 2, also Annex 3 data are required for the precursor formulation. It is crucial that the device settings and parameters are clearly specified, and it has to be ensured that only the intended ESAs amount is generated. In case step 3, the biocidal product is the catalyst containing substance or mixture. In the assessment, there is a focus on the catalyst leaching behavior and on the SOC assessment of coformulants. Generally, no testing of free radicals is required. In case step 4, one has to distinguish if the precursor is a commodity chemical not marketed for biocidal purposes or a non-marketable precursor like seawater or something like that. For precursors that are commodity chemicals, the data according to PPR Annex 2 Title 1 have been submitted and evaluated at active substance approval stage. For non-marketable precursors, such as ambient air or seawater, no data are generally required. Again, device descriptions and settings are required in cases where a device is used. For systems generating free radicals, information on the catalyst has to be given. And also here, generally no testing of free radicals is required. Now I would like to highlight the special aspects of the evaluation of ESAs in the human health section. You already learned that you might cover the Annex 2 information requirements by different approaches. In approach A, the dataset is based on the ESAs. The key requirements are the following. The tested batch has to reflect the worst-case constituent levels of the global composition, and this worst case has to be justified on a case-by-case basis. For very reactive or unstable ESAs, justifications on stability is required. And in addition, an analytical identity profile might be needed. There might be the situation that the tested ESAs batch does not match the worst case. In such cases, the methodology of the technical equivalence guidance is applied. So in Tier 1, you examine if the global composition range is covered, and if not, in Tier 2, you assess the impact of constituent differences on hazard and adapt the data package if necessary. Toxicological reference values are derived in the same way as for conventional active substances. In approach B, information or data is provided for each constituent of the ESAs. Again, you have to include stability justifications and analytical profiles for reactive constituents. It is recommended to explore waiving possibilities. For impurities that are not already identified as ED and are present and are below 0.1%, which is the generic concentration limit as set according to the CLP regulation, in the global composition, no ED data have to be requested, since the identification of such impurities as ED would not have an impact on the classification of the ESAs. With regard to the toxicological reference values, you have to relate and compare toxicity of constituents to the pure active substance. Therefore, you convert the NOAA-EC or NOAA-L values considering the concentration of the constituents. For ease of language, I will now speak of NOAA-L, which is the no observed adverse effect level for oral and dermal routes, but the information also applies to the NOAA-EC, which is the no observed adverse effect concentration for the inhalation route. So we have different cases how to proceed. In one situation, if constituents other than the pure active substance are not hazardous, you base the ESA's NOAA-L derivation on the NOAA-L of the pure active substance. However, if other constituents have lower or comparable toxicity, you base the ESA's NOAA-L derivation on the NOAA-L of the pure active substance and consider the concentrations of contributing constituents. If other constituents have additional toxicity or hazard, then you derive the NOAA-L for each constituent separately. In that case, the risk characterization follows a combined approach. And you also have to consider the mode of actions, which might be different. Here are some examples for the derivation of reference values. In one scenario, there are no hazardous impurities. If the ESAS is composed of 50% pure active substance and 50% non-hazardous constituents, and the NOAL of the pure active substance is 2 milligrams per kilogram body weight and day, then the ESAS NOAL would account for 4 milligram per kilogram body weight and day. This means that you have to add a correction factor to account for the concentration of the constituents. In the second example, we assume that the ESAS consists of 50% pure active substance, 30% constituents of comparable toxicity, and 20% non-toxic constituents. Let the NOAL of the pure active substance be 10 milligram per kilogram body weight and day in our example. Considering a fraction of 80% with comparable toxicity, the ESAS NOAL would be 10 divided by 0.8, which is 12.5 milligram per kilogram body weight and day. And in the third scenario, as said before, reference values have to be set separately and a combined risk characterization is done. Now we come to approach C, the mixed approach, where some endpoints are covered with information on ESAS and others with information on constituents. You have to do endpoint-specific considerations, and you have to transparently describe each dose descriptor for each study. With regard to the toxicological reference values, in the first step, you convert all NOALs to a common basis. So the NOAL is expressed in pure active substance, accounting for the concentrations, similar to the previous example. You do the same for the impurities and express the NOAL in impurity, again accounting for the concentrations. And then you compare the converted values and determine the lowest relevant NOAL for each endpoint. This standardization makes comparison easily possible. However, you do not take those standardized values forward to risk assessment. If you have selected the critical NOALs, you look at the tested item in the respective study or literature. If the lowest relevant converted NOALs comes from ESA study, you use the real ESA study NOAL to base the toxicological reference value on. If it comes from a pure active substance study, you convert it to a calculated ESA's NOAL, considering global composition concentration and base the toxicological reference value on this. However, if it comes from studies performed with pure active substance and studies performed on further constituents, for example, impurities or solvent that cannot be removed without changing the stability, then the toxicological reference values are determined for each constituent. And then you follow the methodology for risk characterization for combined exposure to several active substances or SOC within a biocidal product. In the guidance, there are detailed worked examples for all those cases. In the combined approach, which is approach D, data are available on ESAs as well as on individual constituents for same endpoints. In general, we advise to use data on ESAs to derive the toxicological reference values, unless other information is more robust or relevant. When it comes to product authorization, you profit from the Annex 2 dataset that is already on hand when using a letter of access. For the product part, you perform a, let's call it classical assessment, as for any conventional biocidal product, considering all relevant routes of exposure. And when you think about all those special things that are relevant for ESAs, please do not forget about the substances of concern stemming from biocidal product formulation. They have to be assessed like for any other product. In addition, please also examine carefully if additional Annex II data are needed. This could be the case when you are outside the global composition, or if the active substance contains a precursor that is not specified in a name and was not assessed at approval stage. An example here would be ESA's generation by acidification. So what are the key takeaways? You have flexible assessment approaches. Choose your testing strategy, which is approach A, B, C, or D, based on considerations of ESA stability, available data, and constituent characteristics. Each approach has specific requirements for reference value derivation. Please note that the chosen approach for active substance approval influences the dataset needed for product authorization. The second message is that the global composition is a critical element. We advise to set wide ranges at approval stage to accommodate product variations. Deviations at authorization stage trigger additional SOC assessment and potential further Annex II data requirements. And last but not least, device settings matter. For case steps 2 and 4, precise device specifications ensure proper ESAs generation and risk mitigation for end users. So this was an overview of elements to consider in human health risk assessment for ESAs. And now I would like to hand over to my colleagues presenting the environmental perspective. Thank you.
As a final area, we will cover the assessment of the environment. The information requirements for the in situ generated active substances are similar to the conventional active substances. What is different is that the data requirements for precursors depend on whether the precursor is subject to risk assessment. For the non-marketable precursors, for example air or seawater, normally there is no risk assessment needed and waivers are sufficient. For the other precursors, though, the data are required if the precursor is relevant for risk assessment. You, of course, need to justify it if the data is not submitted. So, how do we identify whether the precursor is subject to risk assessment? We have developed a stepwise approach. So here, first, you need to check whether the precursor is a substance or a mixture, which cannot be itself authorized as biocidal product. If yes, we follow down and check whether the information can be waived, since the precursor is, for example, ambient air or water. If the precursor is ambient air or water, then there is no need for risk assessment and further information. Now, let's go back to the second box in the upper row and answer the question whether the substance meets the criteria of being a substance of concern. Here the acronym is SOC or SOC. In case you handle a precursor mixture, the question is whether this precursor mixture contains a SOC or not. Now here, what I need to highlight is that the substance of concern concept is normally linked to the co-formulants in biocidal products. This is explained in the ECHA guidance on BPR Volume 4, Environment Assessment and Evaluation Part B and C. The identification as substance of concern here, though, does not mean that the substance is considered a co-formulant. Here, I mean in the environmental part and in these flowcharts. What do we do here is that we use these established principles that are clarified in the guidance to identify substances that can cause problems. So whenever you hear in the active substance part us talking about SOC, we do not intend to say that they are coformulants, but it's just the way how we identify a substance we want to have a deeper look on. So now we continue with the third box, and we answer a question whether the substance of concern is fully consumed. If yes, then there is no need for risk assessment and further data. However, if not, we move to the fourth box in the upper row. The question is, does the substance of concern contribute significantly to the overall toxicity of the in situ generated active substance? If it doesn't, then there is no need for risk assessment or for extra data. However, if it does, we move back to the upper row and answer the question in the fifth box. Can release to the environment be excluded, or can it be negligible? If this is the case, and if the answer is yes, then we don't need to do the risk assessment. But if the answer is no, then the risk assessment and the respective data are needed for the precursor. It is a bit complicated. However, following these nice flowcharts, you can come to your conclusions. Now, a similar approach, but with slightly different question, is to be followed also for the reaction byproducts, for the solvents that cannot be removed, and other impurities. In this slide, you see the actual workflow as you can find it in the recommendations, and you find it as figure 18. In the previous slides, we have decided which of our substances are subject to risk assessment. And now we are moving to the flowchart, the figure 19 from the recommendations. And this is a stepwise approach which does not include all information requirements of the Annex 2, but it is a pragmatic approach to avoid numerous data requirements. So what you need to follow what you need to do is to follow for each of the substances that you have identified before, and you will establish which information requirements need to be fulfilled for the substances subject to the risk assessment. For the biocidal products authorization, remember the generic rules that we talked about in the beginning of the webinar. Then you need to examine carefully if additional Annex 2 data are needed. For the assessment of the environment, the classical assessment, as for any other conventional biocidal products apply. You need to consider all relevant exposure, of course. And do not forget about the real substances of concern, meaning the coformulants stemming from the biocidal product formulation. Let me now conclude with the key takeaways messages. In the active substance approval, it is important to choose appropriate strategy, the approach A, B, C, or D, based on the in situ generated active substance stability, based on the available data and the characteristics of the constituents. Then you choose because the approach that you choose for the active substance approval influences quite a lot the data set that is needed for product authorization. Next, risk relevance drives the data needs. Not all the constituents require full assessment. Waivers may be acceptable, but they always need to be justified. And at the product authorization, the consistency with the global composition is key to avoid additional data requirements. And with this, I thank you very much for your attention.
Thank you to all our presenters today. Eva first set the scene on what in situ generated active substances are and how they differ from conventional ones. Andreas has now walked us through the analytical and physico-chemical aspects that make assessment workable in practice. Mari translated that into efficacy expectations under real use conditions. Isabel brought the human health and environmental perspectives together, including what to consider at product authorization. Eva then added to Isabel's presentation with elements to be considered for environmental assessment. I'll now close with 2 short summary slides with key takeaways. After that, we'll move straight to the Q&A, so please keep your questions coming. Now, let me wrap up the core message. In situ generated active substances are fundamentally different from conventional ones. They're created at the point of use, not pre-manufactured and shipped. Now, because of that, we always assess both the generation system and the precursors alongside the substance itself. A second big point is global composition. This is what defines the scope of approval. If your product's output deviates from that approved global composition, you should expect additional assessment and potentially new data needs. Finally, we've shown that the framework is flexible. You can use different assessment approaches, A through D, to tailor how you generate and interpret data based on stability, available studies, and constituent characteristics. The right approach helps you reuse data efficiently while keeping the assessment robust. Those are the essentials we want you to take away. Now a quick word on implementation. Device settings and process conditions matter. They directly influence both efficacy and safety. That's why precise documentation of how the system is set up and used is not just nice to have, it's essential for a sound evaluation and for managing risk. Our final advice is straightforward. Apply clear logic and justify any data gaps. Assess the precursors, the generation system, and the output as a whole, and lean on the recommendations if you need detail on requirements or methodology. Taken together, these practices support consistent, defensible submissions and help avoid surprises later in the authorization process. Now it's time to start our live Q&A session. As mentioned at the start of the event, you can send us your questions through Slido using the QR code on this slide or by going to slido.com and entering the event code that is shown. Our panelists will keep replying until 1 Helsinki time, so do take the opportunity to ask your questions. And as mentioned before, if your question is not answered during the session, don't worry, we'll publish a Q&A document with replies to everything as soon as possible after the event. Thank you all for your participation and engagement today. If you have further questions or would like to stay connected, you can reach us at the contact details shown here, or follow us on our social media channels and subscribe to our updates and podcasts. On behalf of the European Chemicals Agency and all our speakers, thank you again for joining us. We look forward to supporting you in your work with biocidal products. I'll now hand over the floor to Claudio, who will be moderating the live Q&A, as well as our panel of experts, including Christine Hösel-Aguilar for Human Health and Toxicology. We'll start shortly.
Hello. Welcome to the question and answer session of our webinar on in situ active substances. I am Claudio Carlon. I'm the head of the unit dealing with active substance approval in ECHA. And today I will be your moderator. Our panel is made of today's presenter. So I recap, it will be Eva Valkova, that cover the generic part and the environment, Andreas Upov for APCP, Mari Raulio for Efficacy, and we do not have the presenter covering the toxicology, but for that we have Christine Olds Aguilar that also work at the Austrian competent authority. So thank you for submitting your question via Slido. We will reply to all the questions and all the questions within the scope of the webinar and as many as possible in these 45 minutes of live question and answer. But the one that we will not manage, as said, will be replied in writing, and you will find them published on our website. So with the introduction made, we can move to the question. I will pick them up in order of popularity. And the first is the following. For commodity chemicals as precursor, it needs to meet international standard. Is the certificate of analysis sufficient to prove this, or do we need any further analytical tests? This goes to Andreas.
Yes, thanks.
Thank you.
So first, if something can be used off the non-biocidal market, so be a commodity chemical, depends heavily on the approval process. We should assess if an international standard exists and if it controls the precursor sufficiently, and then depends on what this standard is prescribing as proof of compliance, what needs to be delivered. So there are, and I cannot tell in general. It depends on exactly what was decided in the approval. So, and what process is bound to the standard to prove that you're complying with it. I hope that is sufficient.
Thank you, Andreas. Yes, I think that you replied to the question. Let's move to the second. So I have a question for an active substance generated in situ by mixing two precursors. The generation of the active substance requires precise ratio between two precursors in defined condition, like pH. A device is used to control the condition and ratio. Is this active substance considered as type 1 or a type 2? I'm afraid it is also for you, Andreas.
That's okay. We have, I think, a footnote somewhere in the recommendation saying that devices purely used for mixing purposes are not considered devices. This looks like the case would be to precisely define the mixing, so it would not be considered a device. On a side note, it shouldn't make a big difference if this is a device or not. We need to have the exact conditions of the manufacturing process of the in situ generated substance, and if this is done in a device or not, we still need to have the full description of what is happening when the substance is generated. So while I would tend to say it's a case type 1, I also tend to say it actually doesn't make a big difference if it's case type 1 or 2. In any case, you need to describe precisely what is happening when the substance is generated.
Thank you, Andreas. Indeed, it is not always straightforward, but it might not make a big difference. Let's then look to the next. Each device produces a specific composition. We could have as many compositions as devices in the market. to define the global composition, 5 samples within defined ranges is required. Given the large number of different devices available on the market, what exactly should be presented? And Andreas, I think it is still
Yes.
for you.
It's okay. Yes, so we have 2 things we want to know. One is, what is the composition of this active substance? And second, what is the variation? We want to know quite exactly what is the composition of the active substance, and then with 5 batches, and this is the usual target of the 5 batch analysis, and we want to know how does this vary. And I think there is a part in the recommendations saying that you maybe don't need to apply the same methods to the same standard for these tests for the variation, if it makes sense, of course, depends on what variation is. So please test more. It's not to be said that 5 tests are sufficient. We need to know the full variation that is possible with all devices under all conditions. And the 5 batch analysis is mostly what are the details of the composition, and if they change, then maybe you need to do a similar analysis very many times. I hope that is answering the question.
Yes, thank you, Andreas. Indeed, we have to bear in mind the scope of the global composition. So I can move to the next. Do I understand correctly that the status of a precursor as commodity chemical or precursor not marketed for biocidal purpose is determined by how a company puts this precursor on the market? As a biocidal product for in situ generation of an active substance or a generic chemicals? Well, I think this can go to Eva.
Yes, thank you. I can take this one. And the answer is yes. So this is exactly what it depends on. It depends on the manufacturer, whether he wants to place the substance, the chemical substance, on the market with a biocidal claim or without the biocidal claim. And therefore, if it's without the biocidal claim, that means that the way how the in-situ generating system functions, it can use any available substance from the market. But of course, this substance would have needed to be assessed in any case. It would need to be assessed at the active substance approval, or it would need to be then later on assessed in the product authorization.
Thank you, Eva. Very clear. Let's move to the next then. Where can we find the approved global composition? Some active substances have been approved years ago before the new recommendations were available. Good question. Who is taking it?
I start, and Andreas maybe can add later. So yes, indeed, the recommendations have been published in April 2025. And what we, of course, need to say is that they are the best scientific and regulatory knowledge that we could gather on this type of substances. And so, of course, the substances that have been approved before, they would not have the global composition, but they, of course, in their approval would have a certain composition, so people would know what was assessed. And for in the, let's say, future to come assessments, the global composition, if not confidential, will be, of course, available in the active substance approval.
Yes, I don't have much to add. I think actually, this is just a new name for the composition of the active substance. We should have the same level of information about the active substance in approved active substance already and to be approved active substances in the future, which will be all relevant impurities and the active substance itself. And the full composition will likely be confidential, but all the relevant impurities will be published.
Thank you, Andreas. Let's move to the next question on efficacy. So might be for Mary.
Yes, thank you,
But
Claudio.
no, just a moment, because there has been a switch on my screen. So we have another one before in popularity. BPR Annex 2 requirements apply to precursors. For chlorine dioxide generated from sodium chloride by acidification, should a complete BPR Annex 2 dossier, including ED-related studies, be submitted for all the precursor? So, for example, sodium chloride, acids such as hydrochloric acid, sulfuric acid, etc. Maybe this is more for Eva.
Yes, for me. Well, thank you for this question, but we can see that it's very detailed. And so we are, of course, also aware that these substances are right now under evaluation and also under 2 different member states. And the member states are, of course, harmonizing their look on the substances. And so I would like to rather give here a more generic, the generic reply. And then if the applicants themselves would want to contact the ECAs, of course, they are always free to do so. And we can, of course, always, as ECHA, be invited to the meetings if needed. So what I would only reply is that yes, the recommendations shall be followed, and indeed, it in certain cases would require also the ED-related studies, and in some cases not. So therefore, I would leave it like this. Thank you.
Thank you, Eva. And indeed, I mean, we cannot enter into the specificities of specific processes. And the next in popularity, it is the following. It is mentioned for stability studies that even when the degree is low, less than 10%, the degradation product must be identified and measured. What is meant by low degree? Do you have to measure degradation products even if variation is low, like 1%, which can be due to method variation? I think this is more for Andreas.
Yes. Yes, this is correct. I also think this is a bit too open in the formulation to be very helpful. But the idea behind this is that because it's variation or it's degradation of a precursor, it will, or at least it might, have an effect on the generation of the active substance later on. And therefore, you should know, I mean, before the threshold of 10%, because you don't know what happens with the in situ active substance. So for the in situ active substance, we have a presumption that 10% less of active substance doesn't make a difference. For the precursor, this presumption is not really valid because it's not the active substance. And what the effect on the active substance is, we don't know. It can be huge. Therefore, you should know more about the precursor even before this threshold of 10%. And the exact situation for each substance, I guess, needs to be assessed by the ECA at the time of assessment, if there is reasons for concern or if everything is clear. I think that's all I can speculate at the moment.
Thank you, Andreas. I think it was quite enough. The next is indeed on efficacy. Efficacy test should cover the global composition. Dummy products are difficult to be formulated when considering in situ active substances. How should worst case composition be generated to cover the whole global composition range? This is for Mary.
Okay, I've been patient. In general, the worst case is, of course, the lowest concentration of pure active substance. And in case there are other substances that are expected to affect the efficacy, the highest concentration of substance affecting negatively and the lowest concentration of substance affecting positively to efficacy should be tested. And
I could imagine that in very complex cases, some bridging data might be needed in addition. However, I have to say that usually there is some flexibility applied in efficacy assessment of active substance approval, especially for dummy products. But for any details, it would be better to contact the CAs.
Thank you, Mari. So we can move to the next. In general, the in situ active substances are not very stable, especially at higher temperatures. Some parameters like physical hazard, metal corrosion, etc., are determined with tests that last days or weeks at a higher temperatures. How should the in situ active substance mix be tested? Then I think it is really for Andreas.
Yes, it looks like it. Yes, true that in situ active substances are quite often not stable. It depends on the property, really, and it should be the same way waivable as other substances, maybe even easier because of considerations of very low concentrations. I mean, the metal corrosion is an example where you have also instability in conventional active substances. For instance, hydrogen peroxide can quite easily degrade in this test. You will have to make sure that you refill or replenish the test liquid often enough to to compensate. For other physical hazard tests, it really depends on the test. Likely things can be waived, likely things can be tested. It depends. So this question is a bit too generic to be answered, but the same waiving applies as for conventional substances. And I foresee that it should be easier to apply this waiving for in situ substances in many cases.
Thank you, Andreas. And let's see the next. How to set up a BPF for in situ active substances? On which level should the backbone composition concept be applied to the precursor, to the IGS output, the in situ active substance, the dry in situ active substance, or even several of these levels? Well, who is taking this question?
It's on the biocidal product families for the in situ active substances. I don't know if my colleagues would be able to reply, but I think we could rather maybe say that we try to reply in writing.
And one thing is for the biocidal product family, you would have the same composition in terms of coformulants and active substance. If there are coformulants at all, I mean the options given in the question are all different descriptions of the active substance, except for the output possibly. So biocidal products are on the level of output, definitely. And if there's more coformulants present than in just the precursors, then I guess they would be taking part in that. Otherwise, I mean, yes, I'm not even sure we foresee the application of the biocidal product family for in situ substances, but I mean, let's take this offline, yes.
Yeah, thank you, Andreas and Eva. But all the questions will be answered also in writing, so we have the possibility to consult our colleagues. So we can move to the next. How do you define whether the substance concerned contributes significantly to the overall toxicity of the in situ active substance? This may be for Eva, or maybe Christine might want to complement later.
Yes, I suppose this question is asked because I did have it in the presentation, so I assume it might be for environment. And so the substance of concern is significantly contributing to the overall toxicity if it contributes by more than 10% to the overall toxicity. This is what we have agreed in the recommendations, that we would consider it 10%. I don't know if Christina, did you want to add something from human health?
Yes, the substance of concern concept applies when at product authorization step, when some constituents are outside the global composition, and there we follow the guidance like it's laid down in the PPR guidance. So it's if it contributes to the classification of ESAs, then it needs to be concerned that there are other points which needs to be considered, but it should be followed the guidance in place for substance of concern identification. Thank you.
Thank you, Christine. Let's move to the next. GLP labs are struggling to generate a representative version of the in situ substance, as mimicking the real life or in situ process is not always possible in a lab context. The competent authority have little understanding for this, pushing generation of data that might not be relevant or lead to wrong conclusion. It seems more a reflection, a consideration than a question, but I don't know if any of you want to add something or to have a reflection our own. Otherwise, I would take it as a consideration. We take note of it. So we take note of this, and let's move to the question next. Should we expect require a declaration for each delivery of an in situ generated active substance precursor that the supplier of this substance is on the list under Article 95 of the BPR? Maybe it goes to Eva.
Yes, thank you. So I'm not sure I completely would understand the question, like whether is that like a regulatory expectation that that this would be always provided with the precursors. However, so what I can definitely say that in the Article 95 list, you will find the suppliers that are supplying the precursors or the biocidal products. So that would mean that the case types 1, 2, and 3 suppliers would be in the Article 95 list. The case type 4, let's say suppliers would not be there because for the case type 4, the in situ generated active substance is considered the biocidal product. So these suppliers are not on the Article 95 list.
Thank you, Eva. Let's move to the next question then. All our raw materials pass initial SVHC testing, but the candidate list updated biannually. As a recycling company, how do we efficiently verify compliance for the new SVHC in existing material without retesting everything, especially given supplier data challenge? Well, I think this question relates more to recycling and SVHC. I would say it's out of the scope considering our discussion here, but thank you nevertheless for the question. I would give priority to question more in the scope. The next is the following. Is it possible to perform changes on the global composition defined at the approval stage of the in situ generated active substance in the frame of product authorization? Who is taking?
I can take this one.
Eva, thank you.
Yes. So the answer is yes, it is possible to perform changes on the global composition, but of course this is not what we would want to be happening. However, it can happen, but only in very rare circumstances. And one of the circumstances would be that in the active substance approval, there would be the composition or the concentration of the pure active substance of the in situ generated active substance would be in a higher concentration than in the originally approved global composition. And this may lead to the fact that it could be kind of like sent back to the active substance assessment, and the global composition could be enlarged. But this, of course, would only be happening if there is all the available data in the product authorization. So to prove the human health and environment and efficacy. Then the second option that a global composition could be changed or a new global composition could be included into the product authorization is also very, very specific though, and it would be in cases where the precursor that are unidentified in the name, so meaning by acidification, for example. So if in the active substance approval, we would have approved one, let's say, acid, then in the product authorization, you would be applying for a different acid to be included, then this acid obviously would need to be assessed. And in case it will definitely be leading to a different global composition. So then this global composition would be included as a second global composition into the active substance approval. But what we could more or less say here is that if something has been already proven that it works and it's efficacious and it's approved, then maybe in the product authorization that can be sticked to. But we are aware that there are different different IGSs in the all around. So of course there might be applicants that would want to wish to apply for different things, but then they need to be ready to provide the necessary data.
Yes, if I can add to that, I mean, there are a couple of substances where we have these unspecified precursors. These are exemptions. We don't intend to make more of these. This is from the process that we had the description under BPD of the precursors, and what actually was meant was the active substance. And in that process, some of these active substances were bunched together. We don't intend to do this except for those that exist now.
Thank you, Eva and Andreas, to complement. This question, which I highlight first, which approach should be taken when an in situ active substance is generated through multiple chemical reaction? Should the BPR dossier include two different active substances or only the principal reaction? Or would this active substance generated through multiple chemical reaction be seen as a new active substance? Andreas, are you able to address this?
It's a bit cryptical. I mean, we have active substances, pure active substances that can be generated by multiple processes, like we had in the presentation. Peracetic acid generated by 3 different kinds of processes. But I have the feeling this is not meant with this question. I rather read this as two chemical reactions in series. If that is one manufacturing process, I guess it should be described in one. I mean, unless you have intermediate isolation of the intermediate step, then it's different. If it's one, a multi-step reaction taking place at the place of use without further purification, bottling, it looks to me like one in situ active substance. But there is a bit of detail missing to understand exactly what this is about. So I wouldn't want to take a bet on it.
Thank you, Andreas. Let's move to the next. Then about the shelf life of the mixture containing the in situ active substance, what are the requirements? Is there a limit to the shelf life we can claim for an in situ mixture? If a certain shelf life is claimed, is the mixture considered as a good to be stored, requiring specific labeling of the in situ mixture.
I think
maybe it goes to Andreas.
Yes. For the in situ active substance, we don't consider there exists a requirement for storability testing, but we know that some substances are kind of retained before use. So for instance, active chlorine generated to disinfect a swimming pool is quite often used and prepared upfront and then used a day later, a couple of hours later. We consider this part of the generation process, and it needs to be reflected in the variation of the active substance. So if you want to store it for a day, that needs to be, described in the conditions of use, assessed what this means for the composition of the active substance, assessed what it means for the efficacy of the active substance. That all needs to be there. But it's not called storability. But of course, you need to know what happens upon keeping the substance in the place where you generate it for a longer time.
Thank you, Andreas. The next question I see is about efficacy. If a reliable laboratory test following efficacy standards is missing, is it sufficient for an active substance approval to show efficacy through field trials with the relevant precursor, generation system, and condition, even if the impact of pure active substance and other single components cannot be quantified? I wonder, Mary, if you can address it.
Yeah. So laboratory tests are preferable if they are feasible to conduct. So if there is no specific standard method for the intended use, then it would be very good if you could develop something, either amend existing standard method or maybe develop something novel. However, deviations are usually possible if they are very well justified. So a well-conducted field trial might be sufficient. What it comes to the impact of pure active substance and other single components, we don't need to look at that. It's the whole output that we are looking at. Thanks.
Thank you, Mari. Next. If we purchase a generator from a manufacturer supplier who has obtained an authorization to generate an active substance in situ from precursor, will we still need to obtain our own authorization? Quite general. Who is picking it up? From our panelists.
Maybe I can start. It's just I'm not sure we have enough information, so I am assuming that if this someone is a person like that wants to use the generator for their own purposes and disinfect something, then they would not need an authorization. But of course, I would not be exactly sure, like if it's a company that wants to, or like let's say a swimming pool. Andreas, do you have any view?
If you buy a device to produce an active substance with a use description that has been authorized, not the device has been authorized, but the whole in situ generation system has been authorized, then you can use it according to use instructions without having a separate authorization.
Yes.
So I think, and there's also the other case that you buy a device which is not meant to generate an in situ active substance, but you know it does, and you want to use it because you're a big user, and big museums might generate nitrogen from air, and this generator is not meant to kill insects. It's meant just to generate nitrogen for general purposes. You want to use it still, you need an authorization. So both exist.
Thank you. Thank you, Evan and Andreas. Next. Phys-chem parameters with organic precursors with no water as solvent, like dilution stability and persistent form, are scientifically difficult to measure. For example, mixing with the pure water does form 2 layers. How should we comply with the PhysChem request for the biocidal product? Andreas, seems to be for you.
This is mine, and I think there is no specific in situ question here. If it's difficult to test, precursors are not unstable, so that's not a specific problem for in situs. If it's technically impossible, you can always waive. So please apply common sense and normal information requirements.
Thank you, Andreas. Next, when a precursor substance is present in very small concentrations, you can expect probably more than 10% variation due to analytical properties of the test. Can, in this case, higher variation be accepted, providing, for example, proof that the in situ active substance still generates insufficient concentration?
I think that's also for me. Maybe for Mari.
Go.
So maybe Mari can add, but you should aim to be able to specify your active substance with enough precision to know if it's degrading more than 10%. Of course, it might be challenging, yes, I understand, because the concentrations are low, but modern techniques are quite good. If you cannot prove it degrades less than 10%, then you will need to prove that it's still efficacious. But you should aim to be able to prove that it degrades less than 10%. And if you know it degrades more than 10%, you definitely need to show it's still efficacious, and you need to have an idea what is happening.
Thank you, Andreas. So let's move to the next. For the biocidal product authorization, if, for example, precursors are regulated under REACH and information for substance fulfill REACH standard information requirement, but there are information gaps for human health according to BPR Annex 2, is it necessary to generate new information? I would ask it to Christine.
Yes, thank you. Yes, in general, the recommendations foresees that for human health, a risk assessment for precursors needs to be carried out to ensure safe use. And nevertheless, waiving possibilities likewise for normal active substances can be explored. Before going to create further data, it should be clarified with the responsible CA if it's indeed necessary, but it is foreseen in the recommendation that at least Annex 2 data are brought forward and according to the BPR Annex 2. Thank you.
Thank you, Christine. Next question. What are the naming conventions in case an in situ active substance is generated from 2 reactants under acid conditions, same as generated by acidification? I think that maybe Andreas, you can address it.
Looks like mine. Depends maybe on the specific case. So if the acid is there as a catalyst, so not consumed, it would not be considered a precursor, it will not appear in the name, or maybe if it's really important, it might be mentioned under acidic conditions. That would need to be assessed case by case. If the acid is mandatory and is reacting away, I guess it should appear in the name, preferably as a specific acid. As I said, we don't want to create more of these entries that have unspecific precursors, because you don't know what happens if you exchange the counter ion. It might affect the reaction. So it should be done by reacting with hydrochloric acid.
Thank you. Very clear. The next. A biocidal product contains active chloride released from hypochlorous acid. Could you please confirm that a technical equivalence dossier is not required for this active substance, as there is no defined precursor? The BP corresponds to the bottle liquid product obtained at output of electrolysis equipment with sodium chloride and water as inputs. Wow, there are many volunteers.
I can start,
but
and then
Eva
can
was
continue.
first.
So I can start. So from the regulatory point of view, this is not an in situ generated active substance because it says it is released from. So also in the recommendations, we do not touch at all substances that are released to, because those are not in situ generated active substances. And now, I don't know, Andreas, if you wanted to say something else, because in case it was generated from hydrochlorous acid, I don't know if it exists, then we could discuss, but maybe for the sake of time, we only have a few minutes left, so maybe we don't go further. And if the person meant generated, they can, of course, send the question via help desk.
Thank you. Thank you. I see not much to add. We can move to the next. Companies supporting an approval application for in situ substance, still under review, are in many cases generating data for less than 10 years. How will the recommendation affect the validity of this older data, often generated on request of in consultation with the evaluating competent authority, if not in line with the current guidance?
I take that one. So, of course, all the data that has been generated until now, and then especially together, I mean, under the request from the ECA should be taken into account, of course. The new recommendations, or the recommendations, are the best scientific and regulatory knowledge that we could come up with for this type of substances. But of course, we are aware, and we would not want to prolong too much the evaluation of these substances, and we would rather see them submitted to ECHA for peer review, already evaluated. So of course, we would not intend that big, let's say, changes to be done in the applications. But however, saying that, it still, of course, needs to make a scientific sense. So the applicants need to prove that what they are generating and what is then being assessed in their dossier or in their applications is scientifically sound.
Thank you, Eva. I think that we are running out of time, unfortunately. And I'm looking now the question, but I think that we can close here the live session. They tell me that we are toward the end. So I'm afraid that the time together is finished. So with this, I would like to thank you, to thank the panelists. Thank you from my side. I mean, you addressed quite many questions, more than I expected, and thank you. And more work is there to address all the questions that have not been answered, that we will address in writing. But thank you also all the participants following, and the one submitting question to us is very useful for us to provide clarification, but also to understand what are the problems. We acknowledge that the topic is complex. You might like to watch some part of the webinar again, as it will be available on our website. As soon available will be the replies to all the questions, and also the one that have not been answered. So I hope that you found it quite useful and informative. So with this, I would conclude this webinar. Thank you very much from Helsinki. Bye. Bye.